I want to thank Visiopharm for the opportunity to showcase our ongoing research in this forum. My disclosure? Under normal conditions, the stroma acts as a natural barrier against tumor growth. When that balance breaks down, the stromal environment flips to tumor-restrictive to tumor-supportive, a fertile soil for tumor seeds to thrive, as suggested by Paget in his seed and soil theory. Pancreatic adenocarcinoma, shown here in yellow, is a prime example, overwhelmed by a dense fibrotic stroma called the desmoplastic reaction, seen here in blue. This stroma is mainly composed and sustained by cancer-associated fibroblasts and their secreted extracellular matrix acting together as what we call CAF units. Years of research from our group on carcinoma-associated fibroblast biology have revealed a set of key tumor-supportive signaling pathways, spanning from TGF-beta signaling, extracellular matrix architecture, cytoskeleton remodeling, neuronal synapse elements expression, and stromal extracellular vesicles secretion. Individually, each of these has demonstrated measurable impact on PDAC patient outcomes. Together, they reflect the dynamic nature of CAF functional states, capable of shifting the tumor microenvironment toward either a tumor-supportive or a tumor-restrictive environment. As part of an oncology hospital, translating our findings clinically has always been a priority. That opportunity came through Dr. Joshua Meyer, a radiation oncologist at Fox Chase Cancer Center. His work showed that pulsed low-dose radiotherapy, called PLDR, shows significantly reduced toxicity in healthy tissues compared to conventional radiotherapy, called CRT. Evidenced by lower TGF-beta production in healthy tissues, as shown in these mouse colon images. Since TGF-beta is a master driver of desmoplasia in PDAC, PLDR became a compelling bridge between our stromal biology research and a clinical intervention. Working with talented members of the Cukierman lab, we tested in vitro patient-derived CAF units responses to treatment mimicking total neoadjuvant therapy, combining chemotherapy agent with PLDR. Using a novel scoring tool called the Harmonic output of a stromal traits factor, or host factor, we quantify a significant reduction in tumor-supportive activity. The full details are in our recent publication. Please scan the QR code to read it. These results brought research, radiation oncology, and surgery teams together into a phase 1 clinical trial evaluating radiotherapy dose escalation within total neoadjuvant therapy based on PLDR. I'm proud to contribute to this study by characterizing the tumor microenvironment in pre- and post-treatment surgical specimens, applying the host factor to assess whether standard versus high-dose PLDR shifts CAF units activity toward a less tumor-supportive environment. To truly understand the tumor microenvironment dynamics, learning the cell content alone isn't enough. We need to know where these cells are and who they are talking to. The tumor microenvironment biology has now gone spatial. Much like astronomers are mapping the macrocosmos, we can now cartograph the tumor microcosmos, evaluating not just the biomarkers that are there, or the cells that are carrying these, but also the precise location and relationships of every cellular player within the tissue. We can achieve this by using sequential immunofluorescence on the COMET instrument, detecting multiple markers simultaneously to build information-dense images of the tumor tissue. These images are then analyzed in Visiopharm, where AI-trained pipelines like Phenoplex allow us to interrogate each cell's identity and its precise location within the microenvironment. Phenoplex works at a single cell level, detecting and segmenting nuclei and predicting cytoplasmic boundaries for subcellular analysis. It essentially functions like flow cytometry by retaining the spatial context of these cells. By assessing marker expression across thousands of cells within the tissue, it's possible to assign to each one an identity based on the expression profile. For each marker, signal intensity is thresholded to eliminate noise and fluorescent artifacts, as shown in this histogram. Critically, these threshold values are applied uniformly across all samples to ensure consistency. The result? Well, each cell can be unambiguously classified by its unique combination of marker expression. Using Phenoplex, we built a pilot framework to spatially identify host-positive CAF units, comparing normal pancreas against pancreatic adenocarcinoma tissue. As shown in these images, the workflow begins by detecting the tissue area, then moves into single-cell segmentation, and resolves the cellular content within the tissue area. From marker positivity, we can refine each cell's phenotype and quantify population abundance. The comparison here makes something immediately visible, a dramatic expansion of pancytokeratin negative, CD45 negative, Vimentin positive fibroblastic cells in PDAC tissue relatively to normal pancreas. This is the desmoplastic burden I described at the beginning, now spatially resolved. Building on our previous biological findings, which show the coexisting of markers in these CAF units across PDAC progression, we query in this population the presence of double positive cells and corroborated our previous reports. These serve as the rationale to define more complex CAF unit functional states, characterized by multiple expression of additional tumor-supportive traits rather than single markers alone. In this pilot, we focus on a specific tetra-positive CAF unit functional state, host 4 positive, and compare its presence between normal pancreas and PDAC tissue. Using this approach, we identify an increased presence of host 4 positive CAF units within the desmoplastic content of this PDAC tissue. Each yellow dot you see marks the precise location of one of these cells spatially embedded within the tumor stroma. We then use Phenoplex to evaluate neighboring cells within 10 microns of each host 4 positive CAF unit. Interestingly, these fibroblasts were found to be predominantly associated with immune cells, not tumor cells. As shown here, on average, each host 4 positive CAF had 3 immune cells in its immediate vicinity. Drilling deeper, we found roughly 2 B cells, 2 T helpers, and 2 T cytotoxic cells near every host 4 CAF unit. But what stands out is their functional state: low proliferation, minimal granzyme expression, and almost 1 in every 2 T cells were positive for PD-1. These are hallmarks of immune exhaustion, placing host 4 CAF units directly within immunosuppressed and tumor-supportive niches. With our spatial proteomics analysis pipeline established and the clinical trial accrual ongoing, we took a parallel approach, selecting sex and age matched archived cases of patients treated with a CRT-based total neoadjuvant therapy and treatment-naive PDAC patients. We leveraged our biosample repository to construct a COMET-tailored tumor microarray comprising 36 cases to arrange our controls cohort. For this control study, we designed a 26-plex marker panel covering the 3 major tissue compartments: epithelial, fibroblastic, and immune. Markers were selected to interrogate CAF unit functional states and immune subpopulations, with additional markers for cell proliferation and DNA damage to capture therapy-induced stress, which I will detail in the next slides. We then apply our pipeline to treatment-naive and CRT-treated cases. Two examples from each are shown here. To streamline the analysis, our postdoc Sergio developed SPRAN, a Python-based algorithm that characterizes all cell populations from marker expression combinations, quantifies their content, and generates graphical distributions for rapid screening. You can access it via the QR code. These graphs summarize the main cell population shifts between treatment-naive and total neoadjuvant treated cases, suggesting a moderate reduction in tumor cells, an increase in CAF units, and a profound reduction in immune cells. Notably, there is also a sizable population of uncharacterized cells, ones for which our current panel lack markers and worth flagging for future investigation. For CAF unit states, we use a combination of 12 established and novel markers, for which I can only mention their function. Covering CAF functionality, signaling immune suppression, and DNA damage. To focus the analysis, these datasets were filtered using two criteria. One, the phenotypes must be present in at least 0.5% of the total cell population, roughly 50 cells per every 10,000, and detected in at least 10% of the cases in each cohort. These CAF unit states were then classified using a tier-based system ordered by complexity from foundational single functions to maximal integration, where 3 or more functions coexist within a single cell. Each tier reflects an increasingly complex biological behavior within the tumor microenvironment. Two details stood out immediately. First, the treated cohort showed notably less phenotypic diversity than the treatment-naive group. Second, functional states either cluster as group-specific or were shared between both groups, pointing to a common foundational set of CAF units that persists across PDAC regardless of treatment. The common functional states span mid to high complexity, with phospho-FAC, FAP-alpha, and phospho-SMAD2 as the most frequently detected markers. In the graph, the standout difference is in the unknown CAF units, which increase significantly in the treated group. 4 additional functional states also show significant differences. These common functional states are anchored by phospho-FAC and FAP-alpha co-expression, consistent with TGF-beta-driven stromal remodeling and immune exclusion. Additionally, we observe a noticeable enrichment of PD-L1 and CTLA-4 in CAF units, specifically in treatment-naive cases, pointing to an active immune checkpoint-rich landscape in untreated PDAC stroma. Interestingly, we observe a trend toward an increased active TGF-beta signaling in the treated CAF units, consistent with Dr. Mayer's observation in radiosensitive tissue following conventional radiotherapy. Additionally, post-treatment CAF also show evidence of DNA repair activity, suggesting the stroma actively responded to the therapy. Turning now to the treatment-naive unit landscape, CAF unit states were called positive in this group only if they were present in more than 0.5% of the cell population, making them effectively absent in the post-treatment group. Of note, these treatment-naive exclusive phenotypes were evidently enriched in PD-L1 and CTLA-4, besides the foundational phospho-FAC and FAP-alpha, and standing out as the most prominent. As noted earlier, the foundational CAF unit states were further enriched with alpha-SMA and immune checkpoint ligands. Together, they were reinforcing the well-established view of the untreated PDAC microenvironment as highly immunosuppressive niche. In contrast, post-treatment CAF showed enriched TGF-beta and cell-cell interaction signaling, but notably, no expansion of immune checkpoint ligands beyond the already known and shared. This pattern strongly suggests that therapy drives a meaningful reconfiguration of the CAF unit landscape as described in other studies. In summary, we observe meaningful differences in the CAF unit repertoire between these groups. Although this requires validation, given that this control cohort is composed of non-pair pre- and post-treatment samples. Critically, however, both scenarios carry signs of a tumor-supportive niche sustained by TGF-beta signaling. We then apply the same analytical framework to the immune compartment, using a 14-marker panel designed to capture the major immune cell players, their activation and immunosuppression states, and signs of therapy-induced cellular stress. Briefly, both cohorts share an immunosuppressive T helper-enriched baseline. The treatment-naive group layers Tregs on top, while the treated group instead accumulates NK and NK-like CD8 T cells, but these are inactive. Combined with the CAF units landscape, we see two distinct TGF-beta-driven microenvironments, different in composition, but with similar immunosuppressive outcomes, underscoring the need for multi-target interventions. Before concluding, I want to highlight an exciting collaboration with Dr. Haiyan Li. We are integrating our still ongoing spatial proteomics data with tissue neighboring features to train machine learning models capable of predicting whether a given microenvironment is tumor-restrictive or supportive, aiming to build clinical decision tools to guide treatment strategies at the individual case level. Early results from our logistic regression model are promising. Training on the full marker set yields the strongest predictive performance, with an AUC, area under the curve, approaching to 1.0. Notably, CAF unit states and immune subpopulations alone follow closely behind, suggesting that a focused, smaller panel could be sufficient and making this approach far more practical and cost-sustainable for clinical deployment. Finally, to close, I want to say that single-cell digital platforms like Visiopharm are essential for resolving the functional complexity of CAF units and immune crosstalk in the tumor microenvironment. Our data show that untreated PDAC harbors a TGF-beta-driven immunosuppressive landscape, and that neoadjuvant therapy, rather than dismantling it, reshapes it into a different but equally suppressive niche, with NK and subpopulations of CD8 T cells present yet silenced. Both scenarios converge on a tumor-supportive microenvironment, reinforcing the need for multi-target strategies. And finally, machine learning models trained on spatial tumor microenvironment features hold real promises as unbiased, data-driven tools for predicting treatment response and guiding personalized therapy decisions. With this, I want to thank you all for your attention. I want to acknowledge our pancreatic cancer patients and families, and all the people behind these ongoing projects, as well as the funding agencies that have been supporting us. Thank you very much.